Hi micro folks, this is Professor Carberry. This is day 2 of our grape and blueberry fermentation exercise. So you'll recall that yesterday and under 24 hours ago we took some grapes and some blueberries. You'll recall the blueberries were looking like they were starting to decompose a little bit. Oh, did you hear that sound, you guys? I just opened the container. What I did was I put the plastic bags that we had the grapes and the blueberries in. I tried to get as much air out of them as possible. I wanted to create what's called an anaerobic environment. That means no air, no oxygen, because anaerobic conditions will help promote fermentation, right? So if there's no oxygen available, a lot of microbes will switch over to this anaerobic, no oxygen process called fermentation. And you will recall that's kind of what we're after here, is see if we can get some of the microbes that might be on the blueberries or the yeast to carry out a fermentation. And you'll recall that the fermentation that we're really interested in is a fermentation called alcoholic fermentation. This is a type of fermentation that the yeast, a unicellular fungus known as a yeast, Saccharomyces, carries out. And we know that Saccharomyces carries out this alcoholic fermentation. They'll take the sugars and convert them into ethanol, a 2-carbon alcohol, and carbon dioxide. So one sign that we might have fermentation going on is gas production. And again, this morning, guys, I came out and checked this, and when I loosened the mason jar cap, there was definitely the release of gas, and it just happened just then. And I wish I would have held this up and maybe held it closer to the microphone, but definitely as I twisted the lid off, there was an escape of gas. So I think there's a good chance we do have some type of fermentation going on, producing some carbon dioxide. So let me pull these bags out of our kind of secondary container. And again, folks, I was trying to make the conditions inside these bags anaerobic to encourage the alcoholic fermentation. So this one, folks, is our blueberries. This is our blueberries. And the fact that there was gas release when I opened the jar tells me my bags are not airtight. And sometimes what happens when a lot of gas builds up, it'll actually pop the seal on some of these guys. So we could argue, well, maybe we're seeing some gas here, perhaps, right? There'd be better, more sophisticated ways we could see if fermentation was going on, and we'll be exploring those later. Okay, folks, so yesterday what I did was I took one of the mashed up blueberries, and you'll recall that we transferred microbes from the blueberry onto two different types of agar plates, right? And folks, you'll remember that we said the TSA agar plates, which stands for tryptic soy agar, this is called an all-purpose medium, meaning we should have a wide range of bacteria and fungi, including yeast, growing on it. So, so this plate, the side that has our blueberries, is the side over here with lots and lots of growth, right? So that opacity is just all these what we call microbial colonies growing together. They're so close together, it's causing a pattern of growth we call confluent growth, right? It almost looks like frosting. And although I think it's hard for you all to see there, I can tell that there's different types of colonies in that confluent growth, okay? So that tells me that there's a variety of bacteria and or yeast that were on the surface of our blueberries. Now the other side, folks, is kind of fun. The other side was one of the grapes, and I just rolled it over the surface of the plate, again transferring the microbes onto the surface of the agar. And here you get a little bit better idea of how there are individual distinct colonies. You can see some of them are tiny and some of them are big, some of them are more opaque than others. So this is telling me that, again, with tryptic soy agar, that from the surface of the grape we're growing either bacteria and or fungi, potentially yeasts. And with the grapes, you might recall we're trying to imitate maybe how wine production was originally discovered, right? Okay. And then furthermore, folks, what I did yesterday was I took samples of the blueberry juice and grape juice, and I transferred them to a microscope slide, and I'll show you that process here in just a little bit. And I let it dry, and then I heated it over a flame really quickly, just like 3 seconds. And that's to make any of the microbial cells stick to the slide. And then next week, I'm hoping to go into our microbiology lab, and I'll perform what's called a gram stain. We'll stain the microbes present, and then I'll look at the stained smear of our microbes using our light microscope, and I'll work my way up to what's called the oil immersion lens, where we can magnify the microbes 1,000-fold, and that way I can see, if I can visualize the distinct shapes of any bacteria, their arrangements, I'd be able to see if we had any budding yeast present in the juice itself. And again, folks, from our day 2 juice, I'll be making another slide, so I'll show you how that will go. Okay, so obviously this is less than 24 hours, held at garage temperature, and again, yesterday it was like 100 degrees, so we have a lot of microbes present. All right, and then the other plate we inoculated yesterday, folks, was this Sabarod's dextrose media, and you'll recall this media, oops, that's not very helpful. Can we get it so you can see that better? Okay, you recall, folks, the Sabarod's dextrose medium. This medium is selective for fungi, and remember, yeast are fungi. They're unicellular single-cell fungi. And Sabarod's dextrose media, it has a lower pH than the tryptic soy agar, and in addition, it has a higher glucose dextrose concentration. And the low pH and the high dextrose concentrations tends to inhibit a lot of environmental bacteria, but will permit or select for the growth of yeast or fungi. Yeah. So here, folks, again on this side where we have like the confluent growth. Whoops, got this reverse here. This is our blueberries. So we can see we got a lot of possible fungi growing from our blueberries. And then again, over on the other side, this is where we rolled the grape, and you can see there's many of these little tiny, almost like pinhead sized colonies, and I'm hoping those could be wild yeast, Saccharomyces. Yeah, so we're going to be kind of following through with this. So what we'll do today is we're going to replate both the blueberry juice and the grape juice to a new plate, and I'll show you how we're going to try to separate out the microbes so that hopefully we can get individual colonies growing, and that might be a little bit more helpful in trying to identify which microbes are present. Okay, so those are our plates, and you'll notice, folks, I tape them around the perimeter. Right, so it's always a good idea when we're inoculating agar plates. We always, at the very least, want to tape the lid to the bottom in case the plates get tipped over, and that way the lids won't fall off. And furthermore, with some fungi can be really aggressive. And in the micro lab, when students run experiments with the Sabarod's dextrose agar and they have fungi growing, sometimes the fungi, it almost looks like they're trying to push the lid off the plate, right, for those long fungal hyphae. And sometimes they grow in that little tiny space between the lid and the bottom, so they start growing out of the plate, which is a little disturbing. So that's why I think sealing around the perimeter is probably a good idea. Okay, and then, folks, another thing that we did yesterday was using pH papers. I tried to get a rough idea of the pH of the blueberries, and both the blueberries and the grape juice on day 1, they were in between pH 3 and 4, so they're already pretty acidic. Okay, so this is our blueberries, and let me just pull out our grapes here. And because, especially, folks, with the blueberries, since there's looks like a variety of microbes growing, I don't want to do a taste test. And in general, folks, I would really discourage doing taste tests. When I was in college and taking microbiology, my professors would say you can harvest wild yeast, you know, from the air, or you can make this microbial fermentation product or that. And several times in college, I gave myself food poisoning. So I just don't want any of you to get sick. So we'll try to do analysis without actually tasting our items, right? Because we don't want anybody to get sick. All right, so I double bagged this one because it seemed to me that even yesterday it was leaking. These are our grapes. So this is what we're hoping maybe the yeast that we've isolated are Saccharomyces. And I was just trying to see if you could see any bubbles. Sometimes you can actually see some of the fermentation going on, but I don't think this is active enough fermentation to actually see any bubbles forming right now. Okay, so again, folks, what we're going to do, just kind of following through here, what I'm first going to do is I'm going to show you how to transfer the microbes from our grape juice and our blueberry juice onto new TSA and Sabarod's dextrose plates. So this is going to be our day 2 cultures, our TSA plate, and here's our Sab plate for day 2. But this time, what I'm going to do is try to show you a technique. Officially, we call it streaking a plate. And in streaking a plate, what we're trying to do is separate the microbes so that, oops, sorry, and my camera just went off, you guys, so I'm not sure. Let me see here. I just want to make sure that we are still on. Not sure why that happened. Okay, sorry, little delay there. So folks, again, what we're going to do is we're going to streak a new TSA and a new SAB plate, trying to get isolated colonies. And to do this, I'm going to this time use an aseptic technique. And this is going to sound funny, you guys, because when we started with these guys, and they're already so rich with environmental microbes, but this will just give us a chance to practice. So an aseptic technique, sterile or aseptic technique, are techniques we use to prevent introduction of unwanted microbes into maybe sterile surgical equipment, or in our case, sterile media, or if we're trying to isolate perhaps a single type of microbe to get it into what we call pure culture. We don't want all the environmental microbes that are on every surface, even in the air. We don't want them to contaminate our cultures. So we have these special techniques to try to prevent unwanted contamination. So folks, let me see if I can get myself set up here. So I have the source of my microbes, right? And then what I'm going to need, folks, I've got my plates already labeled. And labeling, again, folks, is really important. You want your name, the date. You want the clinical samples, in this case, grapes and blueberries, right? And I also put the type of media, because we're using 2 different media. And then very importantly, you guys, in microbiology, when we're doing sterile or aseptic transfers, what we need to do is make sure the instrument that we use to transfer the microbes is sterile, meaning no living organisms present, right? Including any viruses. So in the old days, we used to use Bunsen burners to sterilize our inoculating instruments. But in the microbiology lab, we had a number of accidents involving like many fires or people getting burnt. So we were so fortunate because last year, the Sac City College budget committee gave us funds to buy what are called back tea incinerators. I don't know if you can see it right here, you guys. Back tea incinerator. And this is a as the name suggests, an incinerator. I've plugged it in, I've let it warm up. And if you guys could see this, if you could look into the barrel, you'd see it's red hot. There, you can see it, right? Red hot, just like an incinerator. And so the beauty of these back tea incinerators is you can still get burnt. So I have to be really careful not to touch the outside of the barrel because it's really hot. But when we switched over to the back tea incinerators, we really reduced the number of fires and instances when students would get burnt. So I really like these. So now that the back tea incinerator, you guys, is red hot, what I'm going to do, this is my inoculating loop, and it's just a metal handle, a thin wire that's been twisted to form a loop at the end, right? And that's why they call it a loop. So this is my favorite transfer instrument. So I need to sterilize it because it's totally contaminated right now, right? And I apologize, my setup here isn't the best. So what I'm going to do is I'm going to insert my loop not all the way in. We've discovered where the wire joins the metal handle. If we stick the loops in too far, that junction, it will experience metal fatigue, and we have the wires just dropping off. And once my back tea incinerator is heated up, it should only take 5 seconds and of my loop inside the barrel, and now everything should be totally destroyed. All cellular microbes, all viruses, even disease-causing prions, you can incinerate, you can destroy them. So to keep my loop sterile, I need to make sure I don't touch anything, right? If I touch something, I have to start all over again, because you just presume if your loop touches anything, it'll be contaminated. And then, folks, you can imagine it's so hot when I pull it out of the incinerator. So you always want to wait a little while, or something that I like to do. I use my sterile auger plates, and I just test my sterile loop to make sure it's not too hot. So I just touch the surface of the auger, and if the auger hisses and spits, that means the loop is too hot. You just hold it there until it stops hissing and spitting, and then you'll know that the loop is cool enough. Okay. And so what we'll do first, you guys, let me see. Let me do the TSA plate first, and then the SAB plate. And let me just go ahead and do the blueberries. So I'm just going to open this up and insert my sterile loop. Oh, it's starting to smell like wine in there, you guys. I think we could have an alcoholic fermentation going on. So that was my blueberries. So what I'm going to do is just spread some of the microbes over a little bit of the TSA plate, and then there's still going to be millions of microbes present. And then I'm going to, with that same sample, I'm going to spread a little bit of the blueberry juice over the SAB plate. And I'm digging into the auger right now, you guys, and I think it's because I'm not very coordinated. So I'm going to put that down. So we just presume, you guys, I've transferred maybe billions of microbes to the surface of the auger plate. Now what I want to do, though, is spread them out, dilute them and spread them out, so that when the individual microbes start growing, making those colonies, those little dots, hopefully we'll get them spread out enough so that they can form what we call isolated colonies. And that's sometimes very helpful in identifying, like it's kind of like the first step of who this unknown microbe might be. But I need to kill all the millions of microbes still on my loop. So what I'm going to do, you guys, I'm just going to put it back in here. 5 seconds. Ooh, you can hear it hissing and spitting in there. All right, I'm going to take it out. And now, folks, what I'm going to do, I'm not going to get any more juice, right? I already got plenty of microbes from the juice. But instead, what I'm going to do, this is the TSA plate, I'm going to spread some of the microbes from where I first spread the juice. I'm just going to pull some of them down. And now I just want to spread some of those guys out over more of the surface area of the plate. Okay, a little bit awkward here. Okay, and then I'm going to put my plate down. I'm going to, now perhaps I have thousands of microbes on my loop, so I'm going to stick it back in the incinerator, kill all those guys, withdraw my sterile loop. And again, folks, this is the TSA with the blueberry. And then maybe in this region, maybe there's like thousands of microbes. I just want to pull some of them down, and then I'm going to spread those out. This isn't traditionally how we would streak a plate, you guys, but I don't have many plates, so I'm trying to spread these guys over half plates, okay? And now maybe I have like hundreds of microbes there, so I'm going to kill those microbes by putting my loop into the incinerator. And then I'm going to pull a subset of those 100 of microbes down a little bit further and then spread them out. And then I'll do that one more time, and hopefully by the end of this process, after we incubate the plates overnight, hopefully tomorrow morning we'll see some actually isolated colonies from that huge gamish of blueberry microbes. Make sure my loop's not too hot, and then I'm going to pull it down and finish the streaking. Oops. Good. Okay. So folks, not to bore you, because I know these videos get really long, so I'm going to repeat that process. I'm going to streak the blueberries onto the SAB, and then I would come back. I want to make sure that I always, before you put your loop down, you guys, always make sure you sterilize it again. So I'm just re-sterilizing it. Okay, alrighty then. Right, I'm going to put my loop down over here. Okay, so I'm going to repeat that process, blueberries to TSA, blueberries to SAB, and then I'll repeat it, you guys, with our grapes. Okay, so it'll be grapes to TSA, grapes to SAB rubs dextrose medium, so we can get an idea of maybe who's the predominant. Like there's probably some competition going on right now in here. We started out with maybe a variety of microbes, and now it's like, well, who's best suited to grow, right, in the grape juice and in the blueberry juice? So we might see over the next week a change in the makeup of the microbial population. So one more thing, folks, actually there's two more things I want to do. I wanted to show you how we're going to make a smear of our grape juice and blueberry juice. Okay, we're going to transfer them to a slide. Kind of mirror image, kind of hard from here, you guys. And on my slide, you guys, I've actually labeled the bottom of the glass microscope slide, and the reason is when we gram stain, there's an alcohol acetone solvent. So very often if we label the top of the slide where the cells are, where the stains are going to go, the alcohol acetone washes it off. So I've labeled the bottom of my slide with the date and with my, I made a couple of circles to guide me where I'll put my samples. So one is blue for blueberries and the other circle is the grapes, okay? And then folks, again, we always want to try to do these sterile or aseptic transfers. Again, I'm going to go back. I'm going to make sure that I have sterilized my loop, so I'm putting it back into the back incinerator for 5 seconds. Okay. And now, folks, what I'll do is I'm first going to harvest some of the blueberry juice. And because it's liquid in here, folks, if I stick my loop in and it's still hot, it will hiss and spit. So I'll just hold it there for a while. And in liquids, usually there's so many microbes present, even if some are killed initially, there should still be plenty remaining. Okay, and then, folks, what I'm going to do is pick up my slide and put that little droplet of blueberry juice on my slide. Okay, maybe just really gently spread it around. Okay. And then, folks, again, not to make this too long, I'll do the same thing with my grape juice. I'll use an aseptic transfer to transfer the grape juice to the other circle. I'll wait for those smears to dry, okay? And then I'll just use our we have a gas stove. I use a clothespin and just hold my slide, my dried smeared slide, just for like 2 or 3 seconds over the flame of the gas stove. And that will fix the cells. That means they're going to stick to the slide, so when I add the gram stain reagent, they won't wash off. It should kill most of the microbes, making the slide safer to work with. And really important, you guys, it's going to preserve the cells. So maybe I ran out of time, or like here, right? I'm going to save my fix slides, my fix smears, and take them into the micro lab next week and do the gram staining. So once I fix them, it also preserves the cells, so I can wait, you know, like a week before I gram stain, right? So that's very cool. And then, folks, the last thing we're going to do is we're going to just re-pH. I am guessing our pH probably hasn't gone down very much. If we had started with something like milk, whose fresh milk pH is around 7, and we introduce some lactic acid bacteria, which will be definitely another project, you guys, we will be doing. Usually after lactic acid fermentation, the milk pH will drop from like pH 7 to pH 4, right? So like 1,000-fold increase in hydrogen ions. But since our fruit juice here is starting out, it's such a low pH, already pH 3 to 4, I'm kind of doubting we're going to see a big change in the pH. So let me just see if I can get this is into the grapes. I'm not doing this very elegantly, am I? Okay, I just need to immerse the four different pH indicators that are at the end of my plastic pH strip, and then, folks, we can see it's turned colors. There's 4 different pH indicators in there, and then the key to this is matching the colors to the colors on the pH box. So this is my key. So I can see here these colors aren't really matching. But let me flip it over here. And again, you guys, this is looking really similar to yesterday. So here the grapes are, it looks to me about maybe pH 3. That's where the colors look pretty consistent. So again, maybe not that big a deal, right? Change. And I'm guessing the same thing, you guys, on the blueberries, that we're not going to see that big a change. And you will recall, folks, since at least with the grapes, we're hoping for an alcoholic fermentation. In alcoholic fermentations, acids aren't made, alcohol is made, right? So we would predict that the pH probably isn't going to change that much. This one's really mucky, but it's looking, I think, pretty similar to yesterday. Not sure if you guys can see that. I think the purple color of the blueberries themselves are interfering a little bit, but it looks to me, you guys, just by that first pH indicator, again, we're around pH 3. So again, I'm going to say probably not that big a change in pH. Okie dokie. And let me see here, folks. Is that all of it? I think that is. So you guys, so what I'm going to do, I'm going to streak the plates again from the juice, day 2 juice. We'll incubate them, see if we can get some isolated colonies. Next week, I'll be doing gram stains, so we can be looking for budding yeast, which would suggest Saccharomyces there, or we can be looking for bacteria, which might appear as little balls called cocci, or they might look like little sausages called bacilli, right? And then I think that the next follow-up, you guys, we might do with this experiment is talk about food poisoning. Remember how I said, you know, if you try this at home, don't eat it because you could give yourself food poisoning. So what we might try to do is kind of follow up with this and say, well, let's say there was an outbreak of some toxin-producing E. coli ON5787, and the suspect is blueberries or grapes, right? How could we identify if an E. coli, which is a fecal bacterium, you guys, so that's always disturbing to have fecal bacteria in your fruits or vegetables. How could we possibly identify whether E. coli was in our grapes or in our blueberries, right? And that will let us talk a little bit more about selective and differential media. Okay, folks, I think that's it for this day 2. And then in another video, we're going to look at another type of fermentation called lactic acid fermentation. And as the name suggests, this is a fermentation that makes lactic acid, right? And it's carried out by a wonderful group of bacteria called the lactic acid bacteria. So we'll be looking at lactic acid fermentation as a way to preserve vegetables like cabbage. All right, folks, so let me get my glove off here. I'm going to stop this video, and we'll see you in the next one.